Okay, so this is my case study 5, GABA as an inhibitory neurotransmitter and vigabatrin mechanism of action. Oop. So starting off with a quick overview of GABA, it is also known, or more formally known, as gamma-aminobutyric acid. It is an amino acid that functions as a primary inhibitory neurotransmitter, all within the central nervous system. So, through the inhibition of nerve transmission, GABA is able to reduce neuronal excitability. Many diseases and psychiatric illnesses have been associated with low and underproduced levels of GABA, and these include general anxiety disorder and diseases linked to symptoms, including seizures and or epilepsy. This is overall due to the decreased levels of GABA, and it overall causes a decrease in inhibition within the cerebral cortex, which leads to the depolarization of cells and hence seizure activity. And there is the structure of GABA. So, moving on to its mechanism of action. GABA works as an inhibitory neurotransmitter within the central nervous system through its ability to lessen nerve cell ability to receive, send, or signal chemical messages to other surrounding cells. In its most spoken form, GABA is a neurotransmitter that slows down the brain through the blocking of specific signals within the central nervous system. There are two different forms of GABA receptors that bind to these nerve cells. The first being GABA A, which is a ligand-gated chloride channel that works to mediate quick inhibitory signals through rapid postsynaptic membrane hyperpolarization. And then there is GABA B, which works to produce slow inhibitory signals through G proteins and second messengers. So moving on to a quick overview of vigabatrin. So it is a medication that is utilized to treat seizure responses and is classified within the anti-epileptic class of medication. It was first formulated in 1974, and testing throughout the years proved that it was very effective in combating seizures and similar symptoms, but they also did discover safety concerns such as peripheral vision loss. So then the FDA approved vigabatrin for treating infantile spasms and refractory complex partial seizures as an additional drug with other anti-epileptic drugs. So while there are still risks associated with this drug, the administration has heavily studied and researched it. So now they are given exact dosages dependent on the patient and their weight, all to heighten these safety measures and hope that this does not occur. And there is its So moving on to the enzymatic inhibition mechanism of vigabatrin. So the main mechanism is that it serves to inhibit GABA catalysis through GABA T, which overall leads to an increase of GABA available within the synaptic cleft to be freed and released in increasing concentrations within the central nervous system. So evidence and research further suggests that vigabatrin possesses the ability to inhibit glial uptake of GABA, and this stimulates GABA released and reduces glutamine cycling through astrocytes and neurons, which overall suggests that there is a glutamatergic effect. So lastly, onto the knockdown of GABA, vigabatrin leads to seizure prevention. So vigabatrin is used to irreversibly inhibit GABA T, which is an enzyme that degrades GABA, and they are both structurally the same, but this comes with an extra vinyl group. So vigabatrin acts as a substrate for GABA T, which allows GABA within the synaptic cleft to be released, which ultimately affects the brain, leading to an increase in concentration that is able to terminate the current seizure activity occurring. Vigabatrin is very important in this process, even beyond its inhibition of GABA T, as it stops the neuronal uptake of GABA, which allows the stimulation of its release to the synapse, overall enhancing the action of glutamine inhibition that adds to anticonvulsion effects. And there is my bond structure of vigabatrin from the Chimera program. And here are my references. Thank you.